The cell cycle can be thought of as the life cycle of a cell where it divides to make two new daughter cells.
During G1 phase, the cell grows physically larger, copies organelles, and makes the molecular building blocks it will need in later steps. In S phase, the cell synthesizes a complete copy of the DNA in its nucleus. During G2 phase, the cell grows more and makes proteins and organelles and begins to reorganize the contents. The G1, S, and G2 phases together are known as interphase.
During mitosis, the nuclear DNA of the cell condenses into chromosomes and is pulled apart by the meiotic spindle. It takes place in 4 stages: prophase, metaphase, anaphase, and telophase. In cytokinesis, also in the meiotic phase, the cytoplasm of the cell is split into 2, making 2 new daughter cells. The ability of cells to have DNA is called genome. Somatic cells are all human cells except the reproductive cells, and the reproductive cells are called the gametes. The clock of the cell cycle is regulated by cyclins, and cyclin-dependent kinases where cyclin is a protein that cyclically fluctuates concentration in the cell. 2 centrosomes are formed by duplication of a single centrosome. Chromosomes are duplicated from S phase and cannot be seen individually since they have not condensed yet. The chromatin fibers become more tightly coiled or condensed. Each duplicated chromosome appears as 2 identical sister chromatids in the centrosome. The meiotic spindle begins to form between the centrosome, which starts to move away from each other. Each of the 2 chromatids each have a chromosome now, and each of them have a kinetochore, a specialized protein structure at the centromere.
The chromosomes in the metaphase cycle have all placed themselves at the metaphase plate with the kinetochores of the sister chromatids attached to the kinetochore microtubules from opposite poles. In the anaphase phase, the 2 sister chromatids separate and begin to move towards opposite poles. Telophase. The nuclei reappears and the chromosomes become less condensed. The division of one nucleus into 2 genetically identical nuclei, which is known as mitosis, is now complete. Then, in cytokinesis, animal cells involves the formation of a cleavage furrow which pinches the cell in 2.
The continuity of life is based on the reproduction of cells, which are also called cell division. Cell cycle control system is a cyclically operating set of molecules in the cell that both triggers and coordinates key events in the cell cycle.
Binary fission refers to prokaryotic cells divided in half into the asexual reproduction of single-celled eukaryotes. Growth factor is a protein released by certain cells that stimulates other cells to divide. Density-dependent inhibition is a phenomenon in which crowded cells stop dividing. A problem occurs when a single cell in the tissue undergoes transformation, which is the conversion of normal cell to cancer cell. The abnormal cells may remain at the original site if they have too few genetic and cellular changes to survive at another site, and lead to the formation of a benign tumor. In contrast, malignant tumors include cells whose genetic and cellular changes enable them to spread to new tissues and impair the functions of one or more organs. Meiosis is a type of cell division that reduces the number of chromosomes in the parent cell by half.